Hi everyone. So today's topic is excretory system, or basically excretion in humans. We are going to discuss about that. So before I start, let me introduce myself. I'm Vani Kari, biology educator at Unacademy. Okay. So I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. So let me just check if this is live. Just a second. Okay. Yeah, all right. Okay, so let's get started then. So we are going to discuss about the excretory system. So specifically how the urine formation occurs, the structure associated with the kidney. Okay, so that is a unit of kidney which is working for formation of urine. So we'll be discussing about that. So let's have a look at the kidneys. So specifically let's have a look at what exactly the kidney parts consist of. Okay, so kidneys are bean shaped excretory organs. So these are bean shaped. So how exactly? So let me just draw here. So let's say this is a kidney now. Okay, so while you draw the kidney, so just make sure that you draw two coverings for this one. Okay, so two coverings. So basically in your exam also they would ask you, or they would expect you to cover draw two coverings as such. Okay, so this is now a capsule which is covering the kidney part. Okay, so now this part where the ureter is there, so let's say the one end of this particular ureter which is this, so this is expanded to form the pelvis. Okay, so this is the pelvis and the ureter will continue down below. Okay, so it will continue down below. Okay, so this is in case of a kidney. So as mentioned here, so these are bean shaped structures. Okay, so this is a bean shaped structure now which can perform lots of functions in your body. So let's have a look at them. So functions of kidneys are basically the excretion and the osmoregulation. So that is maintaining a particular balance. So that is osmotic balance. Let's say water level, mineral level. So all that maintenance is mainly done by the kidney. Okay, so by depending on different proteins again. So but anyway, osmoregulation and excretion are the two main functions when we consider about the kidney. Then kidneys, they help in filtering the blood. Okay, so blood filtration when we talk of, so it is basically the urine formation will occur by filtering blood only. So that is mainly done by the kidney, and urine is actually the filtrator. Alright, so here as kidneys mentioned, so you can see the kidneys are two bean shaped organs. Alright, so let me just draw it here and explain. So while you draw the structure of kidney, again the kidney you are going to show. Okay, so this kidney, the right side kidney, so this one is right kidney. Alright, and you will be drawing the left kidney also. So this is for a complete excretory system. So just make sure that you are drawing the left kidney slightly above as compared to the right one. Okay, so that is because on this side what exactly is present, so there is a diaphragm, the lung, the liver is located. So because of that you can find there is a pressure. So there is slightly, this right kidney is somewhat lower as compared to the left one. So then as usual, as mentioned, so you can see it is 10 centimeter long. So this one is basically 10 centimeter long, you can find. And what else is mentioned? So 6 centimeter wide it is. So this is about 6 centimeter wide. Alright, then located on the urinary bladder. So this is having a urinary bladder. So let's say here the ureter comes, so the expanded end of the one of the ureter, so that is a pelvis. So here also the expanded end is basically a pelvis region where the ureter comes down. Okay, and is projecting into the urinary bladder. So it is projecting down into the urinary bladder here. So this is, let's say this is a urinary bladder over here. So where you can see projecting ureters into this urinary bladder. Okay, so there is an opening here, so that we call as the urethra. Alright, so now as mentioned, so you can see the urine is intermittently emptied. So the emptying of urine is done outside the body, that is through the urethra. So through the urethra, upon the relaxation of sphincter muscles. Okay, so as the impulse is received by the brain, so these sphincter muscles, so these are basically muscles. Okay, so these sphincter muscles are going to relax and the urination can be seen. Okay, so when they relax, only when the sphincter muscles relax, so that is when the urination can be seen. So the urine is intermittently emptied from the urinary bladder to the outside of body, that is through the urethra. So then through the urethra the micturition process or the urination process can be seen. Okay, so now the openings of the ureter into the urinary bladder, so you can see here. Okay, so they are not straight away like this. So what exactly is done? So they are somewhat projecting. So these ureters are projecting, and that means they do not have any valves. So since they are projecting, so they act like a valve. So that is valve, so that they can prevent the backflow of the urine. The backflow of urine should not occur. Okay, so why exactly the ureters are projecting? The ureters are projecting in the urinary bladder so that they can prevent the backflow of urine. All right, so this can be asked. And so when exactly backflow can occur? When this urinary bladder which is there, so if it contracts, so you can see there is a backflow which can occur. So in order to prevent that, so there is basically the projecting ureters you can find. So then the sphincter guards the opening of the bladder into the urethra and relaxes only at the time of urination. So you can see this particular sphincter relaxing only when there is an impulse from the brain is received. Okay, so these sphincter muscles are going to relax and the urination can be seen there. Okay, so let's have a look at the internal structure of kidney. So I'll just explain you how exactly arrangement is done. So first I'll explain this. Let's have a look at the structure, internal structure of the kidney. Okay, so now let's say this is kidney, all right, which is basically bean shaped like I already mentioned. So you need to draw one more covering for this one. So the expanded end of the ureter is forming the pelvis. So let me just show the pelvis also here. Okay, so now this part what you see, so this is basically the pelvis part. All right, so this is the expanded end of the ureter is basically forming the pelvis, and now this is the ureter as such. All right, so now as we already mentioned, so there is one more covering for the kidney. So while you draw the kidney, so just make sure that you are giving two coverings for this, and then you can find there is a cortex region and there is a central medulla region. So now there is a partition for this where you can find there is a cortex region. So now this part what you find, so this would be the cortex region. Okay, then you can find there is a medulla region. So this one is a medulla region. So let me just highlight this part. So we can just see this region what you find here. So this is basically a cortex part. Okay, so this is the cortex region. All right, and the one that you see on the other side, so that is basically the medulla part. So let me just show that as well. So this one is cortex region, and the cortex region you can find it will give a dotted appearance. Okay, so this is basically giving a dotted appearance. So we can show here, so certain dots what you see in this. Okay, so this dotted appearance is seen in the cortex region. Okay, so any idea what exactly is this dotted structure what you find? So what might be there in this kidney which is giving a dotted appearance? Any of you any idea on this? So you can see this dotted appearance here, right? So same way in the medulla region, so you don't see any dots as such, but you can see striations. So stripes you can find. So let me just draw that as well. So now here in this particular region, so that is in the medulla region, you can find pyramids like this. Okay, so this conical pyramids what you see, so they have got these striations. So let me just show. So these are these striations which you can find in kidney. So these are these striations as such. Okay, so this is because of accommodation of nephrons inside this kidney. Okay, so there is nephron which is present. So let me just highlight this as well. So let me just label this first. Okay, So now this part what you find, so this is the cortex region, all right, and this part what you find, so this is the medulla region. Okay, so this goes to be a medulla region, and then you can find now this part what you see, so this is the pelvis region. So let me just label them all. So this is pelvis region, and this junction what you find where the attachment of pelvis or ureter is done with the kidney, so that region or that junction is called as the hilum, and these are conical pyramids. Okay, so these pyramids which are there, so these are conical pyramids, and these are basically renal pyramids. So these are kidney pyramids, and what exactly it contains is, so this particular pyramid which is this, so you can find Henley's loop in there. Okay, so there is a Henley's loop which is present, and also you can find a collecting duct, and that's the reason you can find this particular with the striations or basically stripes you can see. So that is mainly because the Henley's loop is there. Along with the Henley's loop, you can also find that there is collecting duct, and this particular part, that is cortex part. So in cortex part you can find there is Bowman's capsule. Okay, so there is Bowman's capsule you can find, and along with the Bowman's capsule you can also find there is PCT as well as DCT, so which is giving a dotted appearance. So this is the overall structure which you can find, and this projecting end of this, so let me just say here like this, and so this is the projecting end if we mention. Okay, so now this projecting end which is there of the conical pyramid, so this we call as a papilla. Okay, so the projecting end or the apex of this conical pyramid, so this can be labeled as papilla of the kidney. So same explanation is mentioned here. So you can find there is a medulla structure, so you can find the cortex region which is basically darker, and here it is lighter. Okay, so medulla is having a finely striped substance which are arranged in conical pyramids, and apex of this pyramid is papilla projecting into the pelvis of the kidney. Okay, so now the kidney is composed of enormous number of tubules. So this one, as I have already written, so you can find in the cortex region the part of a nephron. So Bowman's capsule, PCT, DCT, so these are all parts of a nephron. The Henley's loop and the collecting duct is also being a part of the nephron. So you can find the millions of nephron arranged here in cortex, as well as millions of nephron arranged where, so they are arranged in the cortex. Okay, so arranged in, so we can just write it here, so these are arranged in cortex as well as medulla region. Okay, so that's the reason it gives a dotted appearance here, which consists mainly of Bowman's capsule, PCT and DCT, and here you can find Henley's loop. Okay, and along with that you can also find a collecting duct. So there is a striated appearance which you can find. Okay, so they have given alternate names, so many alternate names as such. So that is uriniferous tubule is called as nephron, the renal tubule or the kidney tubule. So all these are basically the names of a nephron itself. Okay, so here the structure is given. So while you draw the structure, so like I already said, so just make sure that you draw one more covering, so that is the capsule. So this needs to be done, and as usual you can see there is a pelvis region, the renal pyramid, hilum is labeled, pelvis is region, the medulla, cortex, papilla, pyramid, and this is ureter. So here this one is basically the ureter part. All right, so let's get with the next part. So that is the structure of kidney tubule or the nephron. So how exactly the arrangement you can see, let me just draw it here. Okay, so let's say this is a Bowman's capsule. Okay, so it is just like a ball, so which is pressed on one side. Okay, so you can see it a cup like. Okay, so it is a ball structure. If it is pressed from one side, so you can basically see it to be having a cup shaped structure. So it is just like that. This particular part, the cup shaped structure, what you find, so it will continue below down as the tubule. So you can find there is a tubule as such. Okay, So now this is going to continue as convoluted tubule. So this is a convoluted tubule, you can find it. And then it is going to continue below down here. Okay, so this will continue down, and this is going to form the descending limb. So let me just label each one of these. So now this convoluted tubule is nearer to what? Nearer to this Bowman's capsule. So let me just label. So this one is what? This is Bowman's capsule. Okay, and as you can see, it is nearer to what? This proximal convoluted tubule. PCT is nearer to the Bowman's capsule. Okay, and hence it is this particular convoluted tubule is named as proximal meaning is nearer. Okay, so nearer, so since it is nearer to the Bowman's capsule, so this we call as PCT. So now this is the descending limb. Okay, so this is descending limb, and then it is going to form a loop like this. Okay, so you can find a loop formation, and then it is ascending up. Okay, so this is going to ascend up, so this we call as ascending limb. Okay, so this one is basically Henley's loop. So let me just label this. So this is Henley's loop, and this one is descending limb. Alright, so again it is going to form the convoluted tubule as such. So this is going to form a convoluted tubule as such. Okay, then it will be later on joining the collecting duct which is there. So here you can find there is a collecting duct. Okay, so which is collecting urine from different nephrons as such. So this is going to collect the urine from different nephrons. So this is the collecting duct here. Okay, so let me just label this. So since it is at a particular distance from the Bowman's capsule, so this is called as DCT, distal convoluted tubule. Okay, so now this one is collecting duct. So let me just label this as well. So this is a collecting duct. Now there is a blood supply. So since we already said that, like the filtration of blood, when we mentioned, so filtration of blood can be seen how. So that is when the blood reaches the Bowman's capsule. So at the Bowman's capsule, the filtrate is going to be received. So this is just a cup-like structure which is going to receive that filtrate. Alright, and that's the reason. So here we have to show the arteriole or the blood supply which is done. Okay, so how exactly that happens? So from the dorsal aorta, the aorta which is coming to the back side. So since each of these kidneys are located on either side of the backbone. Okay, so you can find this kidney on either side of the backbone, and the dorsal aorta is dividing further as a renal artery, where it is supplying a blood to the both of the kidney, that is through the renal artery. So this artery which is coming to the kidney, so that is going to branch and rebranch. That means it will go on dividing further to form the arteriole. The arteriole which is entering this Bowman's capsule, so that we call as afferent arteriole. Okay, so this one is what? This is called as afferent arteriole. Alright, which is going to divide into capillaries. You can find there is a capillary formation which can be seen here. Okay, so this is going to form the capillary. So there is a set of capillary formation you can see. Okay, and then this is going to form a specific structure, so that you can find here it is the efferent, it is leaving. Okay, so this we call as efferent arteriole. So this is what? This is efferent arteriole. So now this one, this tuft of capillary, what you find? So this particular part is basically called as the glomerulus. So let me just label this one here. So this is called as glomerulus. Okay, so glomerulus and the Bowman's capsule together, so we call this as Malpighian capsule. Okay, so this is called as a Malpighian capsule. Okay, so they can just ask you to draw the Malpighian capsule in your exam, or they can ask you to draw this nephron structure as well. Okay, so you have to make a note here. The diameter, or basically the lumen of the afferent arteriole, is wider, and here it is narrower. So that is the main reason. So here you can find narrow capillarity, okay, and here also narrow region is there. So that means there is a greater pressure which is generated at this point, okay, and that greater pressure which is generated at this point, so that is a higher hydrostatic pressure which is generated because of the less volume. So there is a great pressure which is generated. So you can find hydrostatic pressure generated due to narrow capillary, okay, and the blood here itself, so that is in the capillary, you can find the maximum filtration or the ultrafiltration process which is happening. So that is blood is getting filtered here, okay. So this process where the higher hydrostatic pressure gets generated at the glomerulus, so you can find filtration of blood occurring, and that process is called as ultrafiltration. So here you can find there is a filtrate which is received, and that filtrate which is received, so that we call as glomerular filtrate, okay. So the filtrate which is received under the high pressure, okay, so that is due to filtration of blood. So this filtrate received in here, so that we call as glomerular filtrate. So this is not the urine, okay. So it is not the urine, it is just the filtrate. So this glomerular filtrate, as we already know, so this contains lots of required things. So there are water, okay, so there might be some amount of glucose, so you can also find sodium might be there in it, okay. So then some amino acids might be there, and other than that the chloride might be there, okay. So then so that is sodium chloride, okay, so that can be there. So lot of required things which will be there. So it also contains urea, the one which we want to get rid of. So in all these cases only the required things are going to be taken back by the blood. So how does that occur? Only the required things can be taken back by the blood, where you can see this arteriole, okay. So this is dividing further, that is forming a secondary set of capillary. It is going to form around this structure, okay. So now it has formed the secondary set of capillary over here, and all these capillaries will join back. Again you can see there is a formation of capillary over here as well, so that is surrounding this part. So you can again see there is a capillary formation surrounding this part which is going to occur, so that we call as vasa recta, okay. So that is secondary set of capillary which is formed here, so this we call as vasa recta. Again all of these are going to join back. So then what will happen? So this will further divide around this DCT as well, okay. So forming the capillary, and then they'll all join back to the renal vein. So this is collecting and again joining back to the renal vein, okay. So that means whatever impurities are received, okay. So let's say this we wanted to get rid of urea, okay. So any excess amount of water, so all the unwanted materials can be got rid of. So that is by this particular system. So where here through this secondary set of capillary, or which we call as, so this is a secondary capillary, or this can also be called as vasa recta, okay. So through which the reabsorption process can be seen here. So here as mentioned, so here you can find the ultrafiltration process, so where you can find, so let me just label this, yeah. So now here as the filtrate formed, so this process we call as ultrafiltration, okay. So this is one process which formed, and same way here there is another process at the secondary capillary. So you can find there is a process which we call as reabsorption process occurs here, okay. So reabsorption process occurs where? So that is at the secondary capillary. or which is also called as vasa recta. Okay, so all the required things like water, glucose, amino acid, sodium, chlorides, so these are all required. So these are going to be reabsorbed back into the capillary, so that is by diffusion. So let me just write here. So this is going to back into the capillary, so that is by diffusion. So this is another important process that occurs while the urine is being formed. So it is a reabsorption process. Okay, I hope this is clear. So let me just check. So like I already mentioned, so you can find a Bowman's capsule, okay, so which I already labeled there. So along with the glomerulus and the Bowman's capsule, so that we call as Malpighian capsule. Then you can find the first convoluted tubule or the proximal convoluted tubule, keeping the Bowman's capsule as the reference point. So this is named as nearer. Nearer to what? Nearer to Bowman's capsule. So as the first tubule or the first foldings what you find, so that is nearer to Bowman's capsule. So it is called as a proximal convoluted tubule. Okay. So the other one what you find, so this one, so this is DCT. So this one is DCT. So DCT you can find at a particular distance from the Bowman's capsule, and hence it is called as the distal convoluted tubule. So I also told you about the secondary capillary network vasa recta. Okay, so how exactly the afferent arteriole and the efferent arteriole. So afferent arteriole is going to enter the Bowman's capsule, and further division you can see. So there is a capillary formation which we call as glomerulus. Then it is leaving, so that we call as efferent arteriole, and then this efferent arteriole is leaving the network. So that is basically it will join the PCT. So surrounding the PCT it is going to form the secondary set of capillary network, so which we call as vasa recta, where it is going to absorb the required things. So since most of the blood content, that is except the cells, okay, and except certain proteins, so you can find most of the liquid part from the blood is actually filtered here at the Bowman's capsule, so that is at the glomerulus, and hence so there are lot of required things which needs to be absorbed back into the blood. So for that reason you can find the water, glucose, amino acids, so all these will be absorbed back or taken back into the blood vessel. So that is through the capillary by a process called as diffusion. Okay, and for that there is a vasa recta formation which can be seen. So any idea like another name for renal tubule, what is it? So another name for renal tubule, so neuron, uriniferous tubule, kidney as well as renal. Okay, so it is given. So any idea? So as we already discussed, so kidney tubule, renal tubule, uriniferous tubule or the nephron, so all of them are one and the same thing. So the Bowman's capsule and the glomerulus, so this is Bowman's capsule along with the glomerulus, so there is a particular term which is given, so that is Malpighian capsule. Okay, so then the reuniting capillaries of the glomerulus. So now as we discuss, so there is a afferent arteriole which is going to divide, so forming the capillary here as such. Okay, so this will go on forming the capillary. Okay, so now this capillaries are going to reunite and they form a particular arteriole. So what arteriole? So this is the one which is entering the Bowman's capsule, so that we call as afferent arteriole. Okay, so the reuniting capillaries over here, so all these capillaries are reuniting. Okay, so it is going to form this structure, so this we call as efferent arteriole. All right, so any doubt till this part? Any of you? So any doubts that you guys have, so you can ask. Okay, so opening of sphincter muscles for micturition. So micturition is nothing but urination. Okay, so opening of sphincter muscles for urination is by the impulse from brain. Yes, that is true. Okay, so only under the impulse from brain, so there is a relaxation of sphincter muscles can be seen. So upon relaxation of sphincter muscles, so basically the urinary bladder is going to empty its content, so that is mainly the urine, and through the urethra the passage of urine will be done. Okay, so it is true here. Alright, so we are done with today's session. So any of you having any doubts, so you can ask. Okay, So thank you everyone. So any doubts you have, so there is a particular channel, so that is my Telegram channel, so Vani Kari ICSE class 9 and 10 biology. You can just search it on Telegram, and any doubts you have, you can get it clarified. So subscribe to our new channel, so that is ICSE and ISC Masters. So click on bell icon to get notified about this session. Like and share the video, and this PDF if you need, so you can just join my Telegram channel, that is Vani Kari ICSE and ISC this thing class 9 and 10. So there you will find the PDF. Okay, so if you need, you can anyway ask me there. All right, so you can subscribe to our channel, a new channel which we have come up with specifically for learners to crack their board exams in a best way. Okay, so see you all in the next session. Bye bye everyone, take care, thank you all.